Okay, thanks, Sam, and it's a great pleasure to be here. Thank you very much for inviting me to this program, and also for giving me an opportunity to speak early on so I can let you know what we are up to, and hopefully this will stimulate some potential collaborations while I'm here. This is joint work that I've done with Young Cal and other colleagues at Virginia Tech. I'll mention some other names later on. But Young was supposed to be here, but he has visa issues and he doesn't dare leave the US. So he won't be joining us. So I want to talk about the cell cycle. First of all, what is the cell cycle? The cell cycle is the sequence of events by which a growing cell replicates all of its components and divides them more or less evenly between two daughter cells, so that the daughter cells inherit the information and the machinery necessary to repeat the process. And all living cells, in order to reproduce, to grow and reproduce, have to go through a cell cycle of some sort or another. And I want to talk about two particular kinds of cells, that is budding yeast cells and an alpha proteobacterium called Colobacter. So let me talk about budding yeast first, and just to explain a bit about the cell cycle of budding yeast. A budding yeast cell divides asymmetrically into a large mother cell and a small daughter cell. The mother cell almost immediately executes an event called start and enters DNA synthesis and starts to make a new bud. And during the rest of the cycle, the bud increases in volume, the bud gets larger and larger, the DNA gets replicated. Eventually, the nucleus migrates to the neck between the mother cell and the bud, and the replicated chromosomes are partitioned into, one set goes into the bud, the other set gets pushed back into the mother cell. And then that process is called mitosis in eukaryotic cells. And after the chromosomes are partitioned, then the cell divides at the mother bud neck, and you get where we started at the beginning of the cycle with a mother cell and a new bud. The bud has to grow a little while before it gets big enough to warrant a new round of DNA replication and division, so buds spend a longer time in G1 phase of the cell cycle before they enter S phase, the phase of DNA synthesis. M phase refers to mitosis. Okay? In the bacterium Colobacter, the cell cycle is quite similar, actually. Colobacter cells also divide asymmetrically. They divide into a larger mother cell and a smaller daughter cell. The mother cell has this appendage called a stalk, and at the end of the stalk is a holdfast, and the holdfast has got sticky glue on the bottom. So the mother cell actually glues herself to a rock or something in the stream and stays put and filters nutrients out of the stream and grows and produces daughter cells. The daughter cells, on the other hand, are born with a flagellum, and they swim away from mama and try to find a place of their own where they can lay down their roots and become a mother cell. So the swarmer cell swims away for a time and then loses the flagellum and creates a stalk at the pole where the flagellum was and sticks herself to a rock and starts to establish her own little area.
Can I ask you a quick question?
Yeah.
How many times can the mother do this? I mean, so it's stuck there.
As far as I know, indefinitely. Yeast cells mother cells don't reproduce indefinitely. They'll reproduce maybe 20 times or so, and then they die. The mother cells, the stalked cells, enter more or less immediately into a phase of DNA synthesis. After they've replicated their DNA, they start to build a daughter cell, so they have a new end and they start to make a flagellum. and the DNA, the two replicated chromosomes get pulled to opposite poles of the cells, and that's called an early pre-divisional cell. And then there's a late pre-divisional cell when it builds a wall and separates between the stalk cell and the swarmer cell and pops off the swarmer cell. The swarmer cells are smaller. As I said, they swim away and they don't do DNA synthesis until after they've carried out this swarmer to stalk transition and become a mother cell themselves. Then they can enter into DNA synthesis. There's no true mitosis in bacteria. Bacteria don't have mitotic apparatus, so we say their cell cycle is a G1S, G2, and then they undergo cell division. Okay, but there are lots of similarities between the bacterium and the yeast cell. Also, lots of dissimilarities. All the common dissimilarities between prokaryotes and eukaryotes. Any questions at this point? Okay, this is the basic physiology of these cells. And I want to talk about the underlying molecular biology that drives these events of the cell cycle. So first let me ask the question, why would we want to use mathematical modeling for studying these cell cycle events? Okay? The first reason is because the underlying molecular machinery of cell cycle regulation in these cells is horrendously complex. This is a simplified version of the molecular machinery that controls progression through the budding yeast cell cycle. There are about a dozen genes in this particular version of the model, and maybe 20 different proteins and complexes, and there are mRNAs. And this is a stripped down version of the budding yeast cell cycle. The Colobacter cell cycle is equally complicated in terms of the number of genes and how these genes are regulated, the protein products, and how they combine to form complexes. And the complexes have various enzymatic activities. They're all tied together in a complex network of chemical reacting species with all kinds of positive and negative feedback loops that make the dynamics complex. And it's difficult, I think, to say the least, to understand and predict what these networks are going to do without some sort of a precise, accurate, calibrated mathematical model.
So you have mentioned that they divide 20 times and then they die.
The budding yeast cells, yeah.
So where is it encoded in the network?
I don't really know, and that's not a problem I'm interested in. The aging of yeast cells, that's a different area. One problem that mother cells have is every time they produce a bud scar. And by the time they produce 20 buds, the whole surface of the cell is covered with scars. That may be part of the aging process. It may be as simple as that. But I'm not going to address that question. I don't know anything about it. Okay? So I think we need mathematical models, first of all, because we want to understand underlying molecular mechanisms, and those mechanisms are extremely complicated. We can't rely on our intuition. We need to have a mathematical model that keeps track of all these interactions. The analogy I like to use is the way molecular biologists do this is like playing mental chess, right? I mean, some people can play mental chess. They imagine where the pieces are on the board and they start moving, you know, rook's queen, the bishop 6 or whatever they say, I don't know. And they play this game, and before too long they're arguing over where the pieces are. It's much easier to play chess with a chessboard and with pieces, and it's much easier to figure out how these control systems work when you have little mathematical representations of all the pieces and how they interact with each other and how they move around on the chessboard. Secondly, the physiology of living cells is extremely rich. So I described the basic events of the cell cycle for these two different kinds of organisms. And every organism is a little bit different. They all do basically the same things. The cells have to grow, they have to replicate their DNA, they have to partition the DNA to daughter cells, they have to replicate all the other components of the cell. They have to control the timing of these events. They have to make sure they occur in the right order. And then in every one of these cells, you can make mutations. Okay, every gene can be knocked out, every gene can be overexpressed, and then you can make double and triple and quadruple mutants. All these mutants have different properties. And if we want to say that we understand how the cell cycle is regulated, we have to understand how it's dysregulated in all of these different mutants. And it's also important for human physiology because human cells are governed by the same kinds of networks, and they work in the same sorts of way in normal, well-functioning cells, but they're also dysregulated in cancer cells. Cancer is essentially a disease of dysregulation of cell growth and division. And if you want to understand cancer, you have to understand how these things break down. Okay, so these are all really complicated and important questions. Important not only for yeast cells and bacteria, but important for us and important for human medicine. Okay. Thirdly, the basic question, I think, is, do our hypothetical molecular mechanisms account accurately for the known physiology of wild type and mutant strains of all of these different cells? In other words, those molecular mechanisms that I outlined earlier, they're hypothetical. They're our guesses of what the underlying control system is, okay? There's no manual that you get with a yeast cell that tells you what the wiring diagram of a yeast cell is. You have to guess the wiring diagram, and you have to guess the values of all the constants that describe the interactions of those proteins. And the only check you have is that your guess, your hypothesis, is consistent with everything that's known about those functioning cells. And then you want to make predictions from those models, and you want to be able to confirm those predictions experimentally. That's the only way we can make progress from a scientific point of view. And this is going to require mathematical modeling. And finally, the mRNAs and proteins in the regulatory network vary extensively in time and space, and their interactions are jeopardized by the random fluctuations due to the small numbers of molecules in these tiny cells. And that's why we need mathematical models to keep track of the dynamics of these systems in time and space, and also to keep track of the stochastic fluctuations, okay? Because the control systems are built to regulate the events of the cell cycle. If those events become dysregulated, it's often fatal to the cell or to its offspring. It's lethal problems. And stochastic fluctuations could easily make these control systems fail. And we'll see how large these fluctuations are and the jeopardy that the control system is in. Okay, you with me? Any questions at this point? Yes,
What's like the range of molecule numbers?
I'm going to get to that. Of course, I'm going to talk about that, okay? I'm belaboring this a little bit because it's a sore point with me, because I've had many papers and grant proposals rejected by ignorant reviewers who don't understand these basic ideas, okay? Both the molecular biologists on one side and the mathematicians on the other side. The molecular biologists say there's no need for mathematical models because they're just telling us what we already know, which is true in a sense because you can't tell those people things that they don't know. And mathematicians say, why do you want to build complicated mathematical models with 50 or 100 equations and 150 parameters? That's not mathematics. Well, I don't know, maybe it isn't mathematics, maybe it's engineering, but I'm not ashamed to be a molecular engineer to try to understand these complicated control systems in detail, accurately, and comprehensively from a mathematical point of view. So if I'm not a mathematician, that's fine with me. I want to understand the underlying molecular biology of these control systems. Now, deterministic models are perfectly fine, I think, to address the question of an accurate account of the physiology of cells, because most of what we know about the physiology of cells are average behaviors over large populations of cells under a variety of different conditions, and deterministic models in terms of nonlinear ordinary and partial differential equations are pretty much sufficient for describing a lot of the experimental data that we have, okay? And I've spent most of my scientific career building and analyzing deterministic models of these control systems. But in recent years, the molecular cell biologists have been able to gather a lot of information on the behavior of individual cells. We can make measurements now on single cells. We can measure the characteristics, the behavior of single cells. We can measure individual molecules in single cells. We can even count some molecules in single cells, as I'll show you. So we now have the data and the information necessary to approach these problems from a stochastic point of view. And the question is, are these deterministic models robust in the face of the inevitable molecular noise in tiny cells? Now, how tiny are they? A femtoliter is 10 to the minus 15 liters, okay? And yeast cells, on average, are about 40 or 50 femtoliters in volume. And Colobacter cells are 50 or 100 times smaller than that, okay? So these are really tiny little droplets of chemistry. And there just can't be very many molecules. As a matter of fact, in a haploid yeast cell, there's only one copy of every gene. There's only a handful of messenger RNA molecules that are transcribed from any one of those genes at any given time. And those messenger RNA molecules are making proteins, and there are only hundreds or thousands of protein molecules of any particular sort in a yeast cell at any given time. I'll give you precise numbers in a second, okay? And it's even worse for Colobacter, because they're even smaller, and of course they have one copy of every gene and a few mRNA molecules, okay? So there is bound to be, it's inescapable, there's bound to be a lot of molecular noise. And our deterministic models don't give us any kind of a description of how big that noise is and what the effects of that noise are going to be on a complex decision-making network, a network that's trying to make decisions, that's trying to govern the behavior of a cell, where making the wrong decision can be lethal, okay? And you don't want random fluctuations in molecular numbers to trigger you to make the wrong decision. Okay, so how many molecules are there? These are some numbers that I got off the web for budding yeast cells, 40 femtoliters. Numbers of molecules per haploid yeast cell for various cell cycle genes. These are cell cycle genes. They encode proteins of the cell cycle control system. I give you numbers of molecules of proteins and mRNAs and the half-lives of these molecules. Now, this data was done by high throughput methods, so we have to be a little bit skeptical, and I'll tell you where the problem is in just a second. Maybe you spotted it already. But if we look at the proteins, that's kind of what we expect. We would expect there to be hundreds or thousands, even 10,000 copies of any one of these proteins on average in a yeast cell. And the half-lives of proteins vary from very short. Cyclin molecules are notoriously unstable. They have very short half-lives, whereas this protein, CDC28, is a very stable protein, has a very long half-life. It's always present in the cells, whereas the cyclin molecules are always being turned over. So there's no problem with these numbers. I think these numbers are probably pretty accurate. The problem is the numbers that are reported for messenger RNAs, okay? Because, you know, when I first wrote these numbers down, I said, these can't possibly be true. There's only one mRNA molecule per cell on average for any one of these genes. And the half-life of these molecules is a half an hour. I said to myself, that can't possibly be right. The control system could never work with these kinds of numbers. Why can't it work with these kinds of numbers? Well, we know that if proteins were made by a simple birth-death process like this, a protein is synthesized and degraded at a rate proportional to its population number, that the distribution would be a Poisson distribution, and the variance of the distribution would be equal to the mean. So the coefficient of variation would be 1 over the square root of the mean, and if there are 1,000 molecules, that would be a 3% coefficient of variation, and that would be no problem, okay? I could live with 3%, and I think more importantly, the yeast cell could live with 3% variation in protein numbers on top of 1,000 proteins. That would be no problem. The problem is that proteins aren't synthesized this way. There's a coupling between transcription and translation. And fluctuations at the messenger RNA level affect the fluctuations at the protein level because the proteins are being encoded by the messenger RNA. And there's a very elegant expression for the coefficient of variation of the numbers of protein molecules in a cell based on the average number of proteins and the average number of messenger RNA molecules and the half-life for message compared to the half-life for protein, okay? And we have all of those numbers in that table that I showed a few slides ago. And if you plug in representative values of those numbers, you find that the coefficient of variation of protein at the protein level should be 70%, okay? And I don't think any control system could possibly work if the protein levels were fluctuating plus or minus 70%, right? I mean, 2 standard deviations would mean sometimes you don't have any proteins, and sometimes you got 2,000 or 3,000 protein molecules, okay? It just couldn't possibly work with those sorts of fluctuations. So those numbers can't possibly be true. Yeah.
Could there be an issue with the fact that aren't the numbers from transient dynamics? And isn't this result from a stationary distribution?
This is a stationary distribution, but no, those numbers are from a.
I mean, they come.
They come from a growing population of yeast cells. You just take an asynchronous population of cells, you grind them up, and you count the molecules, basically. Okay. Yeah. So things are fluctuating up and down, but on average, those are the numbers that were reported.
Yes, maybe what he was referring to is sometimes at the relevant times when the particular molecule is used, there may be 10 copies, and then the rest of the time there's 0.
Yeah, okay, well, that's something you're going to need a mathematical model for to tell whether that's the solution to the problem, okay? And this is exactly why we first started building stochastic models of the yeast cell cycle to try to address this problem and to figure out. I knew the problem was with the messenger RNA numbers and the messenger RNA half-life, so we wanted to figure out, well, what would messenger RNA numbers and half-lives have to be in order for a realistic model to give us the kinds of fluctuations that we see in yeast cell populations? Okay, with me so far? Yeah.
But for this to get this coefficient of variation, you have also a certain model. If you would now say you have the same half-lives and the same thing, and you would introduce some more complex feedback things, you probably could maybe reduce the coefficient of variation in such a model. And this is just like a Poisson, like a mRNA.
Yeah.
Protein.
I mean, yeah, there are questions about bursting and other complicated aspects of transcription that other people have considered. I'm going to show you that I don't think those are really applicable here, that this simple model of transcription and translation with reasonable numbers gives you perfectly good control over the fluctuations in the cell cycle.
Also, Paulson has some inequalities that sort of can be generic with respect to classes of network structure.
Yeah, well, okay. I mean, maybe you can help me out with that later, but I'll tell you what our point of view is right now. Okay, so just exactly how variable is the yeast cell cycle? So yeast cells divide asymmetrically. The mother cell is larger than the daughter cell, and the mother cell has a shorter cycle time. This is growing under ideal conditions with a mass doubling time of about 100 minutes. The mass doubles every 100 minutes. The mother cells are born large and they divide faster than 100 minutes because they don't have to grow as far. The daughter cells are small and they have to grow longer than 100 minutes because they have to more than double in their size before they divide. That's why the cycle times are different. The coefficient of variation of the cycle time is from 14% to 22%. Size at division, average size at division, 68 femtoliters. Coefficient of variation, 19%. Okay. So, they're not particularly fussy about how big they are or how old they are when they divide. What's important to a yeast cell, what's important to any cell, is that you fully replicate the DNA, and then you line up all the chromosomes on the mitotic spindle and accurately partition the chromosomes to the two cells that come out the other side of mitosis. Okay? The duration of G1 phase of the cell cycle is quite variable. G1 is the time from birth until the cell finally decides it's ready to do DNA synthesis and all the other events of the cell cycle. That has a coefficient of variation of 50%, so it's quite variable when they decide that they're going to start DNA synthesis. Okay, so we started building stochastic models of the yeast cell cycle. Most of the work was done by these two postdocs, Debashish Barak and Sandeep Kaur, in collaboration with my colleagues Yang Kao in computer science, Bill Bowman in electrical engineering, and Jean Picot in the Bioinformatics Institute. So I'm going to talk about the model that was built by Debashish Barak. It's based on this molecular mechanism for progression through the cell cycle. This is a simplified mechanism. It really only applies to wild-type cells. It's not complex enough to talk about any mutants, but it does get the cell through the cell cycle from one end to the other with more or less the important components for driving the cell into DNA synthesis and then into mitosis and then into cell division. In this particular model, there's only one gene that's regulated during the cell cycle, and that is the gene that encodes this cyclin called CLIN2. Okay, so this is the only regulated gene in this model. Here's the gene switching between an active form and an inactive form, and that's determined by a transcription factor, SBF. So SBF is a transcription factor that binds to the gene and activates the gene. Once the gene is activated, then it can produce the messenger RNA, and the messenger RNA encodes the protein, and the protein then enters into the protein regulatory network. Okay, that's the basic structure of the model. I don't want to go into the molecular details any more than that, unless you have some questions. Don't. Okay, good. The model is formulated completely in terms of mass action kinetics. Every step is an elementary chemical reaction in this model. There are 58 chemical species, 176 reactions, and 68 kinetic parameters. Okay, they're all first order and second order rate constants for these elementary chemical reactions. And at division, because division is asymmetric, the daughter cell gets 40% of cell size, cell volume at division, the mother gets 60%.
I was just wondering why there are so many fewer parameters than there are reactions. Are you assuming that several of those reactions have the same rate, the same parameters?
Right, because many reactions here, there's an interesting property of these reactions. These proteins are phosphorylated on many sites, okay? So you have a kinase that phosphorylates a protein on 10 or 12 different sites. It's the same kinase. And the phosphatase takes those groups off. And we just assume that those are the same rate constants, okay?
If you have a protein with 10 phosphorylation sites, do you have 2 to the 10 different states?
We do. You don't have to, but this is part of my strategy, okay? So my strategy is I want to do an accurate stochastic simulation of this network, right? And I'm a big believer in Gillespie's stochastic simulation algorithm. It's the only thing I trust. So that's why I wanted to write every step as mass action react, as a mass action step, and I want to just apply brute force Gillespie to the whole damn mess, okay? And I didn't want to worry about tau leaping or whatever these approximations are called for trying to speed it up, okay? Because my attitude at the time, and still my attitude, is that why do I want to make approximations that I'm not entirely confident in to save a little computer time, right? It's no skin off my nose. Computers are good at this, and computers don't give a damn, right? You can let them run all night or all week if that's what it takes. I want to get an answer that I can trust, and I don't have to worry that maybe I got something wrong because of an approximation I made in order to save computer time, which isn't worth saving, okay? Computer time is cheap, mistakes are expensive. That's what I always tell my students. You know, take as much time as you want, okay? Time is cheap, mistakes are expensive.
Question by the same.
Huh?
Untitled parameters, are those fiddle parameters? Are those supposed to be literature parameters?
No. These are guess parameters. These are rate constants, and we don't know what they are. You can't look them up in a book. We have to guess all these parameters on the basis of the data that we have. And I don't want to go into that. People are always asking me where we get these parameters. It's a really boring story, and it takes a long time to explain.
Sorry, so those 58 species, those include all of the components or
Yeah.
phosphorylation
Yeah.
sites? Okay.
Yeah. I mean, that includes all the different phosphorylation steps, okay? And we assume the phosphorylation is sequential. So if there are 10 sites, there aren't 10 factorial different
So it
forms.
simplifies.
Yeah, that simplifies it a lot, yeah. Okay, here's what a deterministic simulation. And now, because it's mass action and I can write down all the equations, I can simulate it as a nonlinear ODE and get a deterministic solution, or I can flip a switch and I can apply Gillespie and get a stochastic simulation.
I just want to make sure I'm interpreting correctly. So you simulate, and then there's a division, and then what do you track after that? A daughter, a mother,
Here
both?
I'm tracking daughters.
Only daughters. The whole
Only daughters here, okay? You could track mothers, you could track them all.
Sure.
And eventually we track them all, we simulate a whole population of cells. Yeah, here I'm tracking daughters. And the rule is, I mean, a newborn cell grows exponentially with a certain mass doubling time. That's depends upon the medium. In rich glucose medium, the mass doubling time is 99 minutes. And the cells divide when CLIB2, which is this blue curve, when CLIB2 drops to about here, that's the signal for the cell to divide. So we stop the simulation. We give 40% of the size to the daughter cells and 60% to the mother cells, and then we pick the simulation back up again.
So how stiff is the system? Like, what's the ratio of time scales for the rates? The worst one you have to worry about?
It's pretty stiff. I don't know what the ratio is, but yeah, you need to use a stiff integrator.
Like how many update events would take place over 300 minutes like this?
For solving the ODEs,
OD, sorry.
yeah, I can't tell you. I
No,
will
no.
tell you for the
Yep.
for Gillespie.
Sorry.
Yes.
So you define it to be dividing when CLIB2 blows the threshold?
Yeah. Because that's the way it works, right? When CLIB2 gets destroyed at the end of the cycle, that triggers a bunch of events called the mitotic exit network, which causes the cell to divide.
So how come we're not seeing discontinuities in the concentrations?
You do see,
Well, there's
a
one
little
of
blip
that one,
there, yeah.
But the other
Yeah.
ones don't
The
seem
other
to-
ones are changing so fast you don't see it, I guess. Yeah, there are little blips when the cell divides. Okay? So that's what a deterministic simulation looks like, and all the proteins are going up and down more or less like you expect them to. That intuitively, this is the way you want it to work. Of course, you have to jiggle the parameters in order to get it to behave the way you expect it to work. Okay.
I guess you don't see, maybe you don't see a jump where proteins go down by half because you're looking at the concentration.
Yeah, here I'm looking at concentration, yeah. exactly right.
Is this all periodic, or is there a difference between these kind of things, or they are just the
Oh,
same?
that's perfectly periodic. Yeah.
So basically, so you can also just look at one.
Yeah, you could just look at one. Yeah. No chaos or anything like that. Okay, so now let's look at a stochastic simulation. Okay, so we just flipped the switch and now we do Gillespie. But I have to tell you, same number, blah, blah, blah. Protein populations on the order of thousands of molecules per gene. mRNAs 5 to 10 molecules per gene. mRNA half-lives on the order of 2 minutes. So we have to set those parameters to get things right. And then a Gillespie
What
simulation
is the
looks like
total
that.
number of molecules in the system?
Well,
I want to do just particle-based simulator, so how many?
yeah, so there are about a dozen mRNAs, so the rest are proteins. So there's 45 proteins with 1,000 molecules each, plus assorted mRNAs, yeah.
50,000.
Okay, so these are the kinds of fluctuations that you can expect, I think, based on a Gillespie simulation. They're bigger than 3% because of the transcription-translation coupling, but they're not 70% because of the number of mRNA molecules and the short half-life of the mRNA molecules. So you get some temporal averaging over the mRNAs. And the system works very reliably in the face of this kind of noise. The cells almost never die, but the timing of and the events of the cell cycle occur in the right order, but the timing of the events is quite variable, but it agrees with what you see experimentally, okay? So here we have the experimental results and the model results for the total cycle time, for the duration of the G1 phase of the cell cycle, and for size at birth, okay? And the means all agree very well with the experiments. That's because the parameters in the model have been adjusted to get the mean values right, and you can do that using the deterministic model. But what we're really interested in the stochastic model are the coefficients of variation. And they're all in reasonable agreement with the kinds of variability that you see experimentally. I mean, the worst one is this 6% difference here, 5% difference there. Yeah. Any questions? Now, there's a lot more known about variability of individual events in the yeast cycle, but I didn't want to go into all those details. We don't really have time to do that. But there's a lot more that could be said. Here are histograms for G1 times in mother and daughter cells. They look pretty reasonable. And here are histograms for the messenger RNAs in the molecule. And most of these histograms are Poisson distributions because almost all the genes, the messenger RNAs are driven by just simple birth-death processes. The only one that's regulated is CLIN2, and that's the only one that's not a Poisson distribution. It's pretty close to 2 Poissons, but I'll talk about that more in a sec. And remember, I mean, these histograms were computed before we had any data. We didn't know what the numbers of messenger RNA molecules were for cell cycle genes. I guessed that they were between 5 and 10, so we set the parameters up so that they would be in that ballpark. And those levels of mRNA molecules are enough to control the fluctuations at the protein level, enough to give me the kinds of variabilities that you see in the physiology of yeast cells as they progress through the cell cycle. If the means were much smaller, the variations would be much bigger. If the means were much larger, the variations would be smaller than observed.
Maybe a naive question, I've lost track again. So these, at what time are these taken, right? So you're going
This is
through
for
a cycle.
so we simulate an asynchronous population of cells, and then we just take a random sample of cells from an asynchronous population spread all throughout the cell cycle. That's exactly the way the experiment is done, so that's the way we simulate it. Okay. But for the mRNAs, that's not so important because there's no regulation over mRNA
Except for the
synthesis, except for the CLIN2. Yeah, exactly. Okay, so we set out to count mRNA molecules because about the time we were doing these calculations, we learned about this new technology called FISH, fluorescence in situ hybridization, which allows you to label and count individual mRNA molecules. You label them with little red fluorescent dots, and every dot here is one mRNA molecule, okay? You're looking at a single molecule. So here's a yeast cell, here's the bud. It's almost ready to divide. The blue is the staining of the nuclei. And as I said, every red dot is an mRNA molecule. So this cell has 3 mRNA molecules. This cell has 1, 2, 3, 4, 5, 6, 7, 8, 9. This one has 4, okay? And you just go through and you count mRNA molecules per cell and construct the histogram, okay? That was the computed histogram, what we guessed it would be. That's the histogram that we later measured for this NET1 gene. They're almost identical. That was luck. And that's the best fitting Poisson to the data, okay? So these really are Poisson distributions. That's why I think a simple birth-death process is a perfectly valid approximation to how messenger RNAs are made in these cells. Okay, the only problem is with CLIN2, because CLIN2 is regulated, and we didn't get this quite right. That's what we expected the histogram to look like from our choice of parameter values. That's what we measured the histogram to look like. That's the best fitting Poisson, which isn't any good at all. But you can fit this to 2 Poissons, a Poisson down here and a broad Poisson up there. And that's a very good fit, the 2 Poissons, as if the gene has 2 different states, an on state and an off state. And now we have the data, we can reparameterize the model and get the CLIN2 expression almost right on, okay, by just changing some parameters in the model a little bit.
Can you do some statistical tests that the 2 Poisson fit is not overfit?
I'm not that good a statistician, so I can't answer that.
And when you changed these parameters, did it keep the cell
Yeah,
cycles
everything else
about the
was
same?
fine. Yeah. Okay. Now, some mutant strains exhibit what's called partial viability, a phenotype that's inconsistent with deterministic models. Because in a deterministic model, a cell is either alive or dead. There's no such thing as a cell that's maybe alive or maybe dead. Okay? But there are some mutants that have that property. In particular, this famous strain where these 2 B-type cyclins have been mutated so that the CLIB2 cyclin is extra stable and the CLIB5 cyclin is gone altogether. And here's what the experimental data looks like. The plus sign, these are 3 different experiments in each case. 3 experiments for wild-type cells and 3 experiments for mutant cells. And we're counting the number of cells as a function of time on a semi-log plot. And you can see the wild-type cell number is increasing exponentially at a certain rate. Okay? It's a straight line on a semi-log plot. That's a wild-type cell. This green curve is a wild-type strain, and this blue curve is a wild. They're all growing at the same rate, and that is just the mass doubling time of the culture. The mutant cells, however, are the solid dots, and here's one mutant strain, and you see the number doubling time is considerably less than it is for the wild-type cells, although the mass doubling times of the culture are the same. Cells are increasing in mass, but they're not increasing in number, and that's because some of the cells are dying or leaving the cell cycle and not creating progeny. Okay? And here are simulations of the situation, a simulation of a wild-type culture growing and a simulation of the mutant cultures growing with a smaller number doubling time. Okay? So this is many realizations of a wild type culture growing exponentially in time, and you see that these cells are growing in galactose, and in galactose the mass doubling time is 148 minutes, and the number doubling time is 151 minutes. It's a little bit longer than the mass doubling time. That's perfectly fine. Whereas in mutants, if we do many realizations of the mutant culture, they're all growing with a longer number doubling time of about 190 minutes, and that's the kind of dispersion that you see in the number doubling times of different realizations of the mutant culture. Okay, and what's happening here? What's happening here is easier to see if you plot the, what's this called? This is the opposite of a cumulative distribution. This is the probability that the interdivision time is longer than t. Okay, so for wild type cells, you see there's a certain minimum interdivision time, and then the probability that you're even longer than that drops off very rapidly. Okay, and all the cells are dividing, right? This goes all the way down to 0. There aren't any cells that don't divide within 250 minutes or so, because all the cells are dividing. But if you look at the mutants, a bunch of mutant cells are dividing, and then it levels off here because some of the cells never divide. Some of the cells get stuck in the cell cycle and never divide. And that's because this mutant cell is kind of close to some kind of a bifurcation. If it's on one side of the bifurcation point, it can go through the cell cycle and divide. If it's on the other side of the bifurcation point, it can't get through the cycle, it gets arrested, and that's like an absorbing boundary. Once it's arrested, it can never divide. Okay? And essentially about 40, in the simulation, about 45% of the cells get arrested and never divide. That's why the number doubling time is considerably longer than the mass doubling time. Okay? Any questions?
So they're still healthy in a sense?
They're alive, but they can't divide, and eventually they'll die because they get bigger and bigger and they'll die of obesity or something. Okay, so let me switch to Colobacter and see how far I can get in the second half of the talk. Okay, so Colobacter is a bacterium. It has these two forms, the stalked cell, and the swarmer cell. Okay. I would like to talk about two different parts of the molecular biology of these cells. Maybe I'll only get through one of them. The simplest story is this protein called POP-Z. POP-Z is a marker. It's a marker of the poles of the Colobacter cell. And a newborn swarmer cell has POP-Z polymers at the swarmer end of the cell. And the POP-Z polymer is attached to a protein that's attached to the chromosome. So the chromosome is kind of anchored to the old pole of the cell. The old pole is the pole with the flagellum. The new pole is where the cell divided. Okay, when it divided, that's called the new pole. Okay? And the swarmer cell has to grow for a while and then decide to drop its flagellum and make a stalk. So that's the transition to becoming a stalk cell. And about the time of that transition, we get a new focus of POP-Z polymers at the new end of the cell. And the DNA gets replicated and the new chromosome gets pulled over to the new end of the cell by the POP-Z at this end. And then this FITZ-Z protein moves to the center of the cell, and the FITZ-Z ring is the ring that's going to cause the constriction and cause the cell to divide, okay? So POP-Z is involved in coordinating the ends of the cell and the replicated chromosomes and the FITZ-Z ring. And it's important that it always stays at the old end, and halfway through the cycle or so, you get a new locus of POP-Z polarization at the new end of the cell. And if you prevent the cells from dividing so that they grow longer and longer and longer without dividing, that's called a filamentous cell, you will see loci of Fitzsy polymerization along that filamentous cell with a certain periodic distribution. Okay, that's the clue as to what's happening here. Okay, so here's our model. We think this is a Turing type instability from that result for the filamentous cell. You see loci of POPC polymerization at with a kind of standard spacing. Suggests that there's some sort of a Turing instability here, but you have to modulate it a little bit to make it fit the facts, okay? The modulations are, well, first of all, it's a substrate depletion kind of a Turing mechanism. Remember, there are two kinds. There's substrate depletion and activator inhibitor. This is a substrate depletion. The substrate are the POPZ monomers, and the activator is the POPZ polymer. And the POPZ polymer autocatalytically turns on its own polymerization. The polymerization process is autocatalytic. That's what makes it Turing-like. Autocatalytic for conversion of monomers to polymers. Of course, the monomers diffuse faster than the polymers do, so you satisfy the basic condition for a Turing instability. But you need to have a de novo pathway for polymerization as well as the autocatalytic pathway for polymerization. And finally, we need to include coupling between gene transcription and protein translation in the model. If we put all these pieces together, then we can explain the pattern of POPC polymerization. So here's the first calculation. The cell is growing. You can't see it here. Can you turn the light off for just a second? Maybe it's not worth figuring out how to do it. You can see it here. This is the boundary of the cell. So the newborn cell is about 1 micrometer in length, and the cell at division is about 3 micrometers in length. Okay? So the cell is growing, and in this case, we don't allow the gene to replicate. So if the gene doesn't replicate, the gene is sitting here close to the old end of the cell, and the gene makes messenger RNAs. The messenger RNAs don't diffuse very far. They're big, bulky molecules, and the messenger RNAs encode the protein, and the protein just kind of polymerizes at the old end, okay? And you don't see the new locus of POPC polymerization. But as soon as you include gene replication, okay, so this is about the time when DNA synthesis starts, and now you get a new chromosome, and the new chromosome is pulled to the opposite end of the cell. So now the old chromosome is sitting here, the new chromosome is sitting here. There are 2 POPZ genes, so they're making messenger RNA at the old end of the cell and at the new end of the cell, and now you start to get a new locus of POPZ polymers at the new end of the cell, once the cell gets big enough to support 2 regions of POPZ polymerization. Okay? That's the characteristic Turing wavelength for the pattern. You with me? Okay, so that's the basis of these POPZ patterns, and okay, and these, as you can see, these are stochastic simulations of the model. Okay, we also have, of course, have deterministic simulations that look really smooth, but what I wanted to show you were the stochastic simulations.
This is your RDME type model, or
It's a reaction diffusion. What's the end part?
master equation? It's like spatial Gillespie method.
Spatial Gillespie. Yeah. Okay, now this is only a tiny part of the whole mechanism that gets you through the cell cycle in Colobacter. Okay, it's module 1. How you get the POPZ polymers at the 2 ends. Now, that triggers a bunch of other events that are described by all these other modules and blocks. We know how we want to modularize the system, but we haven't done it all yet. Only about half of this stuff has been put into mathematical forms and kind of hooked together, okay? And the reason I'm so sore right now is because we submitted a renewal proposal. We thought we'd make good progress on this. We tried to renew it, and the reviewers just decimated it. They say, why do you want to build a whole bunch of equations to describe this big, complicated system? That sounds really boring to me. Well, maybe it is boring to you. I don't give a damn if it's boring to you. It's interesting to me, and it's important to biologists to understand this whole control system. Okay, I'm just a little angry. I think you. Just let me vent, okay? So here's what's happening. The POP-Z is localizing to these two different poles. POP-Z recruits a protein called SPMX. SPMX recruits a protein called DivJ, and DivJ is the input to module 3, and module 3 ultimately controls the phosphorylation state of this protein CTRA, and the phosphorylation state of CTRA determines whether the cell is going to be able to replicate its DNA or not. Okay, so this is a really important chain of events. And so here's what happens. A newborn swarmer cell is full of the phosphorylated form of CTRA, and it can't do DNA synthesis. When it undergoes the swarmer to stalk transition, DivJ gets recruited to the new pole, and DivJ sets off the chain of events that clears the cell of the phosphorylated form of CTRA. And now the cell can do DNA synthesis, okay? So it goes through DNA synthesis, goes to the early pre-divisional stage, and POP-Z appears at the new pole. And then POP-Z, let's see what happens. DivJ, okay, yeah, I'm a little confused because this is where it gets confusing. And it's not particularly intuitive anymore. The way this system works itself out, DivJ causes the phosphorylation of DivK. DivK inhibits DivL, right? And that causes CTRA to become unphosphorylated. But anyway, you get a gradient of phosphorylated CTRA across the cell. It's more phosphorylated at the swarmer end and less phosphorylated at the stalk end. And then when the cell finally divides in half, you only have phosphorylated CTRA in the swarmer cell. So you come back to where I started. Whereas in the stalk cell, you don't have phosphorylated CTRA, so it can go immediately into DNA synthesis again. Okay, so this is a crucial distinction between the swarmer cell and the stalk cell. And it all has to do with the movements of these proteins between the two ends of the cell, and then the reactions and diffusions that are occurring at the end and through the whole body of the cell, and then cutting off the diffusion when the cell finally divides. Okay, so it's a complicated process. It's important in coordinating the events, and the only way to figure out how it works that I know of is to build an accurate mathematical model of the reactions and diffusions and localization events that are occurring in the cell and simulate it accurately from a stochastic point of view. So let's see. This is a deterministic simulation. You can tell by looking at it, this is a deterministic simulation. DivJ gets localized at the old pole at 30 minutes, and then it triggers a whole sequence of events which clears out the phosphorylated form of CTRA in the middle portion where you can do DNA synthesis, and then phosphorylated CTRA comes back at the swarmer end of the cell, but it's very low at the stalked end of the cell. And then when the cell finally divides here in the middle, you get the two different states, okay? And here's what the various realizations of phosphorylated CTRAP look like. In the swarmer cell, it's high, in the stalk cell, it's low, and in the pre-divisional cell, it comes back high again. And then these are stochastic simulations of all of these processes.
What makes
It's robust.
The DivJ is the most upstream thing.
The DivJ is the most upstream thing.
And I can see you're holding it at the pole, right?
Yeah,
So
yeah.
you're kind of forcing the gradient.
Yeah, we're
So
forcing
what
it.
is it? What's at the pole?
Well, POP-Z is at the pole, and POP-Z recruits DivJ to the pole. But why doesn't DivJ go to the other pole when POP-Z goes to the other pole? I'm not quite sure, okay? But we don't have a full model of the whole system yet.
How is, do you know the min system?
Yeah,
Is pop something like min, or is it a little different?
I don't know it that well. It's not like min. Fitz Z is a lot more
Because
like
min, they get
min.
these
Yeah, I
oscillations.
know.
It's very
that.
similar to the Yeast. Does not have the MIDI removal, but it has the self positive feedback loop there.
Yeah, sorry, yeah.
Yeah.
But that's a different bacterium.
Yeah, that's a different bacterium, a different story. Okay, you'll give me 5 minutes? I just wanted to mention a couple technical challenges then. And Young should be giving this part of the talk, not me, so I might say some foolish things. But one problem is stochastic simulations of the transcription-translation coupling, okay? Because you only have 5 molecules of the mRNA, but you have thousands of copies of the proteins, so it could be kind of awkward from a computational point of view. And the question is basically, you have this big range of mean number of molecules in the cell and propensity of the reaction. Some reactions occur many, many times, and other reactions are very infrequent. And you want to build a hybrid kind of a solver, and where do you have to do Gillespie simulations, and where can you do ODE simulations? That's the basic question. And so for our model of the budding yeast cell cycle, it was a simplified model, and it has about 25 different reactions, and we just did a brute force Gillespie simulation and counted the number of firings of each reaction as a function of the lowest average molecule number of the limiting species for that particular reaction. Okay, and it's all over the place. Here are some reactions that are very infrequent and have very few molecules. Those are the dangerous ones. And here are reactions that have lots of molecules and fire lots of times. Those are the ones we should be able to simulate with ODEs. The question is what to do with these guys, okay? And what we have discovered is we can get a pretty accurate hybrid simulation if we just do Gillespie on the reactions that are down in this corner, and we use ordinary differential equations for everything else. Okay? And it was described in this paper in the Journal of Chemical Physics. We tried 4 different strategies and full Gillespie on the whole thing. And what you can see is strategy A is too limiting. It doesn't give you accurate results. It's really fast, but it doesn't give you accurate results, okay? Strategy B is pretty darn fast, and it gives you very accurate results, okay? It's 5 times faster than full Gillespie, but gives you exactly the same results. Okay, just by doing Gillespie on the few reactions down here in the corner and solving everything else by ODEs.
So when you say you solve by ODEs and then couple it with this, and maybe this is the question for Yang. So
Yeah,
email him, but
Yeah.
so you have partial counts then, right? You're solving an ODE. So then how do you, I mean, do you round when you're doing the stochastic version?
We can talk about
Okay,
that.
yeah, it's not.
I know, I can maybe give you an explanation, but of course you can find it in the paper. Okay, second thing is how do you optimize your parameter values in a stochastic context, okay? So you have stochastic simulations, you have to do many, many simulations, but then you also have, the data that you have is statistical data. So you're trying to fit stochastic simulations to statistical data, and that's a real pain in the butt. But we found that this procedure called quasi-Newton stochastic optimization actually works pretty well. In the one case that we tried it worked. It really surprised me how well it worked. I didn't think it was going to work, but it did. But now we have to try it on some more challenging problems to see whether this is really going to be a method by which we can optimize the parameters in stochastic models with respect to realistic statistical data that we collect on dividing cells. And then stochastic simulations of reaction diffusion equations. There's a number of issues that need to be addressed. First of all, it's a growing domain. So it's not, you don't do a fixed discretization of the domain, but that problem was basically solved by Crampon, Gaffney, and Maney some years ago, and we just used their approach. But some reviewer of one of our grant proposals challenged us on this and ticked me off again. We also have fast diffusing species and slow diffusing species. And for the slow diffusing species, you have to use a fine grid. But if you use a fine grid and you use Gillespie, you spend all your time watching the fast molecules move back and forth between the little grid spots. 99% of the calculation doesn't get you anywhere, okay? And multi-grid methods have proven kind of useful in getting around that problem. That's pretty standard. And the other problem is trying to simulate nonlinear chemical reactions in small voxels. And Sam knows a lot about this. He can help me on that regard. But basically, when you have a bimolecular or worse, a trimolecular reaction, and you're trying to make it go in a small volume, you just don't have enough molecules there to do anything, so nothing happens, okay? And basically, you have to smooth the concentrations over a number of voxels, and you have to be really careful how you do that to get an accurate result. And here's just an example of the phosphorylated state of CTRA in Colobacter. When we did the deterministic model, the deterministic model's got a lot of it in the swarmer cell and just goes to 0 in the stalk cell, and then it comes back in the pre-divisional cell. That's what we expected to do. And when we just tried to implement that dumbly in a stochastic simulation, it just didn't clear CTRA out of the cell because the degradation reaction became so infrequent and unlikely, it just didn't happen. And then when we corrected that and did some smoothing, then we could get the stochastic simulation to look a lot more like the deterministic simulation and like what we expect is happening in the cells. Okay, so here's my summary. Cell cycle regulation in both prokaryotes and eukaryotes is a fiendishly complex problem that will be sorted out only in the context of accurate, comprehensive mathematical models, stochastic models. Secondly, although neither molecular cell biologists nor applied mathematicians are particularly enamored of accurate, comprehensive mathematical models, they better get used to them. It's the way life is. These models give coherent and accurate accounts of the basic physiological properties of proliferating cells, both prokaryotes and eukaryotes. The control systems seem to be operating at the limits permitted by molecular fluctuations in femtoliter-sized cells. And stochastic versions of the models are perfectly consistent with detailed quantitative measurements of cell cycle variability in time and space. So here are some references, and if people have some questions, well, I'm happy to talk about this. And of course, other people can leave anytime. Yeah.
Thanks. So I have a question for you regarding the fish technique you showed us. It seems like when an mRNA molecule is not regulated, you see a nice Poisson distribution.
Yeah.
And this can give you insight on the modeling to a large extent, because in your model you have only one messenger RNA that was regulated, which was I think Queen 2 or something.
Queen 2, yeah.
But that was almost a modeling choice you made. You could have chosen some other proteins, because in reality there's regulation, you would think everywhere. I mean, there's lots of regulation. So by doing these simple fish experiments, you could determine which proteins are actually regulated, right? And so I wanted to ask you, have you compared the distributions of other proteins in your model with the fish?
So there are two ways to tell whether a gene is regulated or not. You know, the classic way, the high throughput way, is you measure the mRNAs and you can see whether they're going up and down during the cell cycle with mRNA technologies, right? And so we actually, you can look it up on the web on databases whether they think these genes are temporally regulated or not, okay? And CLIN2 turns out to be one that everybody knows is temporally regulated. NET1 is one that everybody knows is not temporally regulated. Okay, so there are easier ways to figure it out than what you just suggested. Okay, but our fish experiments are certainly perfectly consistent with the other way of doing it. Okay, the model I showed you, as I said, was a simplified model. We only put regulation on CLIN2. We know CLIB2 is regulated, so the CLIB2 calculations are not correct because the CLIB2 gene isn't regulated, and as a matter of fact, it is. We have a new version of the model, which is 2 or 3 times more complicated than the one I talked to you about, and it has 7 genes that are temporally regulated during the cell cycle, and 5 or 6 of them are regulated. So it has many more genes, many more proteins. We still solve it by brute force Gillespie, but it's now complex enough that we can address the properties of mutant cells. And in fact, the mutant that I talked to you about is described in the new paper. And the new paper hasn't even been submitted for publication. Okay. Yes.
So beginning, you said 1 mRNA per cell was the high throughput result.
Yes.
What was wrong with that?
Yeah. It's interesting, right? So in the high throughput result, you grind the cell up, and then you just assay every different mRNA because you have the full genome, so you can pick out all the mRNAs. So actually, what they got right are the relative amounts of mRNAs relative to each other. In order to get the absolute amounts, they had to guess the total number of mRNA molecules in the yeast cell. And where they come up with that number, I don't know, but they were off by a factor of 6. It's as simple as that. And if you just change that total number of mRNA molecules, then your numbers are completely consistent with our fish numbers and other people's fish numbers.
So the denominator.
It's the denominator.
Normalization.
Normalization error, yeah, Sam?
But there's also an issue with the half-lives too.
Yeah, the
You used a much
half-life.
shorter one in the model.
Yeah. So how you measure half-lives is trickier still, okay? Because the way you measure a half-life is you put in an inhibitor of mRNA synthesis. So you don't make any new mRNAs, and then you watch the mRNAs go away, okay? But when you block transcription, you block the transcription of the genes that are encoding the proteins that degrade mRNAs. So it's not obvious that you're getting the right. So there are better ways to measure mRNA stability, but not high throughput ways.
I feel like this is the 3rd time I've asked the same question, so I'm just going to ask it
Yeah,
again.
go
But I
ahead.
seem to not be able to get it. So if you have empty set to P for some, or M for some mRNA, and then degradation,
Yes,
right? So you get a Poisson distribution
Yes.
at stationarity.
yes.
But you have a model where I don't know, maybe it goes to station, I don't know, but it goes up, right? It is whatever it is, and then you cleave it. Like the daughter cell, you lose 60%. So you go down, and then you come back up, and then you cleave it and go down. So if you grind all those up, I would not expect, well, I know I won't see a Poisson. So even though the model is Poisson, it's this halving every so often. So you should change the distribution, no?
Well, at some point, the number of genes doubles. And the gene gets replicated, and when the new gene starts to be transcribed, we don't know.
Dave, if you kill every existing molecule with a 40% probability, wouldn't it give you Poisson still? Like instead of making it exactly 60%.
You're just saying you're thin.
Yeah. And what if, I mean, especially if you have a compartment where it's not gonna be exactly 60%, but just, you know, I mean, everything has a 60% chance to be in the mother's compartment.
But wait, I mean, none of that is dependent on how big the cell is, because we're just looking at number of cells. And I'm not saying the probability that an mRNA degrades is proportional to volume. Whether the cell is growing or not doesn't make any difference.
No.
It's being synthesized at a fixed rate,
But then there's the killing part.
and it's
There's the-
disappearing at a constant rate per
Yeah.
unit time.
So that, no, I agree, that gives Poisson. It's the at some point in the model, you take away 60%.
Of the volume, but what difference does that make? The new cells, each one has one gene.
No, don't you divide the number of of mRNA molecules also?
Yeah, okay. How do we do that?
Do you like one for each one or something?
I guess we partition them.
Yeah, that's what I'm talking about, not
When
the,
we partition them 60/40. Okay, I'd have to look at
okay.
the code.
I mean, how long does it take to reach equilibrium again?
Yeah.
So
Depends
that's a
on
short
the
amount of time.
rate.
Depends on
that's
the
true, that's
half-life,
true.
right? Yeah. And the half-life is pretty short, right? It's a 2-minute half-life compared to a 100-minute cycle, so that should come back to steady state pretty fast. And I think we'll have to look at the data in the new model, but I think you can see a little dip in the unregulated mRNAs when the cell divides, and it quickly comes back to the steady state number of molecules. Maybe that's the answer to your question.
Maybe we should take the rest of the discussion offline since we're over. All right, thanks, John.